Thank you, Surana, for the introduction. My name is Farhan. I'm from the MNI. Thank you all for coming. Today I will be talking about how we decipher neural signals by dissecting visual system hierarchy. So at the beginning of the talk, I'm going to tell you what neural signals are, what they represent, and then what is a visual system hierarchy, and then finally how to decipher these signals by dissecting the hierarchy. So what are neural signals? So this is what a recording looks like that we obtain. It consists of spiking activity, which are all or none action potentials. It's sort of like a binary code, as we see here. And it also, that shouldn't be there. But anyway, it also consists of something called local field potentials, which are continuous voltages. These are analog voltages that we obtain, and we can divide these into different frequency bands and analyze the data that way. So we have a raw signal. We have two types of signals, spikes and local field potentials. Now, what do they represent? It has been known for decades that spikes represent the outputs of neurons, whereas the local field potential represents the total synaptic activity occurring at the tip of the electrode. And local processing that is happening within the circuit is reflected in both local field potentials as well as in spiking activity. Now, why is it important? So we know what spikes are, but as far as the local field potential goes, it's sort of ambiguous and it's not very clearly understood today, because we don't know if local field potentials are more consistent with feed-forward input into a circuit or with the local processing occurring within the circuit. And that's important because the bold signal that we obtain using fMRI imaging studies correlates with the LFP, the local field potential, more than it does with spikes. So it's important to understand what the local field potential represents. And it's also important for studies of neural prostheses because they also employ the local field potential to decode the signal. So again, this is what we want to know. This is the topic of the study. Are local field potentials more consistent with the input or with local processing? So how do we do that? We use a technique known as macaque in vivo electrophysiology, in which we have a monkey sitting in a chair who looks at a fixation spot, and we stimulate the neurons in his brain by using different types of visual stimuli. And we obtain a signal that looks like this. Now, where do we record from? How do we do this? So the three areas which are part of the motion system hierarchy that we looked at are known as V1, MT, and MST. V1 is the primary visual cortex, is the area in the cortex which receives visual signals first. It projects to area MT, and then area MT projects to MST. And here's a schematic of what this hierarchy looks like. So here we have feed-forward signals from one area to the next, and we also have local processing that is happening within the area. And what I did was I recorded from each of these levels of the hierarchy to understand and to answer this question. So again, the question is, how are spikes and local field potentials related to each other? And do local field potentials reflect feed-forward input or local processing? So again, that's the question that we're looking at. So the stimuli, let's see if this works. So the stimuli that we use to actually do the study this is what is called a grading. So these are motion stimuli that actually trigger activity in these neurons. And here you have a grading, for example, moving down and to the left, and then this is moving to the right, and so on. On the right here is what is known as plaid stimuli. This stimuli is actually composed of two gradings on top of each other. And again, this stimulus goes to the right, and so on, and it goes around the circle 12 in 12 different directions. So this is what the monkey is looking at. So just one thing about how these stimuli are actually constructed. So here's an example of two gradings. So here's the grading moving down into the left, and this is moving down into the right. And here you see a plaid which is moving downward. And you see the plaid as a coherent motion which moves downward, which is not consistent with either of the two grading directions. So this is a coherent perception that we see. And these are the stimuli that we looked at to answer these questions. So how do we classify? So we use these stimuli, we use gratings and plaids, and we try to classify neurons in these areas. So how do we classify them? So here's an example of a neuron which prefers downward motion. So it has a preference for downward motion stimuli. Here are 3 plaids. The green arrows represent the directions of the individual grating components, and the red arrows represent the coherent motion of the plaid as the plaid is moving. So there are neurons in the brain that are only able to detect the individual components of the grating. They're not able to actually see the coherent motion that we all see. And these neurons are known as component selective neurons. And this is what their response looks like. It selects only when the grating is moving in the direction that it prefers. Then there are neurons which actually integrate the responses of the low-level neurons and can signal the coherent motion of the plaid as we all saw it. And these neurons are known as pattern-selective neurons, and this is what their tuning curve looks like. And as you see here, the tuning curve is very similar to the tuning curve of the grating. So this is how we classify these neurons. And you can think of these as smart perception cells because they correlate with perception more than the other cells. So we have, this is how we classify the neurons. Now, what do we get in the brain? How does the activity look like for the entire population in these areas? So here's the schematic again, areas V1, MT, and MST. So here what I'm showing is, on the y-axis, patternness, so to say. It's how much pattern is the neuron. And on the x-axis, I'm showing componentness, which is how much component is this neuron. And each dot represents a single neuron. Blue dots represent component cells, red dots, pattern cells, and black dots, those that cannot be classified as either type. So in area V1, we see that most cells are component cells, which makes sense because it's a very low-level area, and it doesn't integrate information as high areas do, and it can only see the individual components of the plaid. Area V1 projects to MT, and here we see that there's a greater percentage of pattern cells in MT. Now, these studies had already been done by previous labs, so we replicated them, as we see here, and we found what was found by those labs, such as Tony Mofshin's lab and other labs that are out there. So what was new in our study is what happens in the next area, in area MST. Nobody knew. And we expected there to be an even higher percentage of pattern cells. And that's what we found. That most cells in MST are pattern, and this makes sense because it's the highest area in the hierarchy, and it's supposed to have more cells that correlate with perception. So this is all fine, but how can we, the initial question, how to determine if the local field potential is more consistent with feed-forward activity or local processing. So this is when we address that issue. So here's the schematic again. Now what we saw in this highest area, MST, is that area MST rarely contains component cells. And the local field potential, which is a sum of activity of many neurons. And in MST, the sum of all these pattern neurons is unlikely to be component. So if we can find a component representation in the local field potential signal, then it is likely to be feed-forward. Because locally in MST, it's pattern, and the summation of that is going to be pattern, most likely. So thus, any local field potential signal that is component is coming from somewhere else. It's coming from somewhere upstream of MST. So this was the question now. Is the local field potential component or pattern? So here's an example site in area MST. This is the response to a grating, and this is the response to a plaid of an MST neuron. And we see pattern selectivity. Both responses are identical. When we look at the local field potential, so here I've divided into high gamma, which is very high frequencies, and low gamma, which is low frequency, bands. We see that, as we see in blue here, the response is pattern. But the low gamma response, which is red, is component. You see the bilobe response. Now this indicates that for this site, the local field potential for the low gamma frequency band is consistent with feed-forward activity because it's component, based on our hypothesis. Now we wanted to see what happens across the population. So this is data for area MST. So again, I'm plotting patternness and componentness. We see that there's a preponderance of component selectivity across the frequency spectrum in area MST, except for the high gamma where we see some mixture of pattern as well as component selective responses. So this makes sense because area MST gets most of its input from component selective neurons. It gets it from MT, it gets it from V1, and so on. Now, if we were to look at MT, we would hypothesize that mostly all of the local field potential signals should be component because MT gets its input from V1, which is mostly component cells. And we found what we expected, that most sites in MT are component selective. So next I will tell you about an observation that we made in our study. So here what I've done is I've lined up the high gamma response for MT and area MST with the spiking responses of area V1, MT, and MST. And what we noticed is that if we shift this response by one area, we get something that the high gamma response in MST resembles the MT spiking response, and the high gamma response in MT resembles the V1 spiking response. So this is what we get for high gamma. When we look at the low frequency LFP, local field potential responses for low gamma, and if we shift by two areas below, we see that the MST low gamma response resembles the area V1 spiking response. So it's just an observation. It's nothing, it's just something we noticed. So in conclusion, the percentage of pattern neurons increases along this visual system hierarchy. Most MST neurons signal pattern motion and are rarely component selective. And thus, to estimate the feed-forward input, we wanted to look at the component response. And we found that in area MST and in area MT, the component responses are prominent across the frequency spectrum. And so we propose that the local field potential is heavily influenced by feed-forward sensory signals from lower areas, particularly at lower frequencies. And low frequencies are consistent with feed-forward input from distant sources, whereas high frequencies are consistent with feed-forward input from more proximal sources. So again, the initial question, what do LFP signals represent? What are they more consistent with? We found that they're more consistent with feed-forward input into areas. And so this is just some ongoing work that I'm doing at this time. What we're doing is we're actually injecting a drug called musomol in area MST to inactivate the activity. And once that happens, we're trying to see if the local field potential is, again, component. Because now we've managed to inactivate the local activity happening in MST, so now we would be even more certain if we found a component response in the LFP that it represents feed-forward input into this area. Thank you for your attention.
Thanks, Farhan.